So starting the basic introduction, what are variants? So the number of reads that align at a given position in depth or coverage. So what is depth or coverage? When analyzing the NGS data, we get the reads file. So a read is called a depth or coverage. So variants are simply deviations from the reference sequence. The thing that deviate from our reference sequence, like cytosine site is changing into thiamine and adenine in our reads file, as you can see in the illustration here. The cytosine is changing into thiamine and thiamine is changing into adenine, and all the other variants are remaining same. So that is a short nucleotide polymorphism site that is identified by the genomic analysis by a pipeline. So these are variant. And moving on, what are some kinds of variants? There are SNPs, single nucleotide polymorphisms, in also known as SNPs, and the single nucleotide variation, SNPs and SNVs, in individuals. Like if you are dealing with a lot of organisms, they would be called SNPs, and when you are dealing with a single individual, they will be called a SNV. So these variants are simply exchange of one nucleotide base pair for another. For example, cytosine is changed into adenine, adenine is changed into thiamine, just like that. And there will be Indels, short for insertion and deletion. These are added and subtracted base pair in a segment of DNA. It is estimated that humans have several million of insertion and deletion sequence variations. CNVs. CNVs are these refers to the difference in number of specific gene for a certain trait found in a genome. For example, while the central dogma taught us that there were two copies of a gene in every genome, there could be three, there could be four. These are the changes that we defined in CNVs. So these are also the variants type, also called copy number variation, single nucleotide polymorphisms, single nucleotide variation, also insertion and deletion, and copy number variation. So a question that you should ask right now, how does NGS data allow to be so much confident that SNPs and Indels can be identified from this kind of data? So answering your question, although NGS yields a large amount of data of sequencing, its ability to transform its medicine, genetic medicine, relying on identifying relevant variants with the high confidence. So variant identification begins with the alignment of NGS reads to the reference genome. For example, if here is written human reference genome, for example, you are not using the human variation technique. You are using some other kind of technique for your data. You are using the organism of Saccharomyces cerevisiae. You are using plant, want to identify plant variations. So that was so explaining this with all. Here you can see there is a reference genome sequence line. If I can just point my laser onto it, here you can see here is the line for my reference genome. And here are the NGS reads. These are the depth, 3x, 5x and 8x depth. Here you can see there is single nucleotide polymorphism. And here is insertion and deletion. So these are the types of variations that occur in NGS data. So these could also be homozygous reference, heterozygous, homozygous alternative, and these other kind of stuff. How they can occur in a human file, how they can change one human to another. So these are type of variants that we usually deal with. So what basically is variant calling? So variant calling is a process by which we identify variants from sequencing data. Here in the illustration, you can see there is an SNPs sequence that is identified, and SNPs is now related with the gene, and then it is distributed across many genes. Then thousands of SNPs are then identified in the chromosome, and then a SNP profile is constructed of all the chromosome to check how many single nucleotide polymorphism were involved in formation of that NGS data. So how to do all kind of this stuff, you just have to carry out whole genome or whole exome sequencing to create a FASTA-Q file. We just have to create a FASTA-Q file. After FASTA-Q, we have to align the sequence to the reference genome just by creating a BAM file. So we will create a BAM file. After creating a BAM file, we will identify the aligned reads differ from the reference genome and then write it into a VCF file. So we will create a VCF file as well. After getting to know, you should have know what FASTQ is. So FASTQ, I should explain. FASTQ is a format that we use where we get our reads from the sequencing companies. And BAM is the BAM binary alignment map that we use from the same file, a sequence alignment map where we contain our sequences, aligned sequences to the reference genome. And after that, what is VCF file? So VCF file, I'm gonna explain you to this course. VCF is a preferred format because it's ambiguous, scalable, flexible, allowing extra information to add it into many fields. So you should know what VCF is. It's a variant calling format. So it's a variant. There are it's a tabbed eliminated data file. It contains the chromosome, its position, its ID, its reference, and how it was changed. Like thymine was changed into adenine, quality, its filter, its information, format, and a lot of other things that are now processed in a VCF file. So many of million variants can be stored in a single VCF file. So that was for the all that was here for our VCF file, how VCF file are created and how to do all this. We just have to get the FASTQC. After getting the FASTQ file, we just have to then align the file and create a BAM file. After aligning the file, we have to extract our variants and then create a VCF file. So it's a 3-step procedure as well. So that was it for the whole introduction lecture of this variant calling course.